Okay, so if I remember correctly, we got up to here last week, right? So I'm going to take it on from here. And basically what the program is going to be today is look in a bit more detail into the lytic replication cycle in Bacteriophage T4. Well, I don't know. First, the kind of attachment entry step, then the lytic replication in Bacteriophage T4, lysogeny in Bacteriophage lambda, and have a little look at the impact of phage on human health. And then for the rest of the time, I want to just, well, we'll go on to like human viral infections and COVID-19, okay? So that's quite a lot of stuff, so I'll try and move quickly, okay? Right, so this is kind of just a summary of where we got to last time. You know, you have to bear in mind virus replication and cell division are two fundamentally different processes. You have to remember how the plaque assay works. And in order for any virus to replicate, it's got to complete each one of these steps in the lytic replication cycle. Okay, so the first part is attachment, specifically binding to the right host cell. Viruses are adapted to a particular host. And if you're a bacteriophage that infects Escherichia coli, there's no point in binding to the surface of Mycobacterium tuberculosis, because all the rest of your replication cycle will not work. So any virus, any bacteriophage has got to bind to the right cell. And this specific binding is mediated by the interaction between a phage attachment protein and a receptor on the host cell. Okay, so first, we're thinking mostly about I won't go too much in detail about this. So like for Bacteriophage T4, it's like kind of like, it looks like this, right? So the attachment protein are the proteins at the end of these fibers, okay? So it's here is the attachment protein. For Bacteriophage lambda and this phage SPP1, it's the proteins that are at the end of the flexible tail. So that's where the binding is going to occur for these tailed phages. Now, what about the receptors? Well, of course, they're going to be structures that are on the outside of the bacterial cell. For gram-negative bacteria, a lot of phages bind specifically to the O repeats on LPS. It can be a receptor for phage. Or they use outer membrane proteins, like this one, OMP-C, OMP-C, outer membrane protein C, which is a porin, or LAM-B, which is the permease for maltose. Some phage use pili, okay? The conjugation pilus is used as a receptor by phage M13. So all of these are structures in gram-negative bacteria, but of course, there are many phage which infect gram-positive bacteria. Now, because the cell wall structure is very different, that means that phage infecting gram-positive bacteria also have to use different types of structures for the entry receptors. Now, I don't know a lot about this, to be honest, but this is one I looked up, okay? So this is a phage that infects Bacillus subtilis, gram-positive bacteria. And UEB is a large transmembrane protein which crosses the thick peptidoglycan layer. So it's got part of the protein is really on the outside of the cell wall. Okay, so whatever the receptor is, whatever type of bacteria, it's got to be a structure that's right on the outside, okay? Because it's the first place where the phage can really get a grip on the host bacterium. Okay, once the phage binds, the next thing it's got to do is get its DNA across the peptidoglycan layer. So Bacteriophage T4 does this by releasing lysozyme and drilling a kind of hole through the peptidoglycan through the plasma membrane and then will inject its DNA into the host cytoplasm. So, okay, I'll show you a kind of video about this. Actually, no, I'm going to leave this, okay? Well, you can check it out on your own if you want. It's a nice video, but basically it's the same thing, okay? So, the principal point here is phage lysozyme can punch a hole in the peptidoglycan. And that allows the phage to inject its genome into the host cell cytosol. Okay, once the phage DNA is there, yeah, thank you. Okay, so the phage DNA, very first step, phage DNA gets injected into the host bacterium. It will circularize, and then you'll start to have the expression of early phage genes. So one of them in T4 will be phage DNAs, which will degrade the host cell DNA. Another early gene is phage DNA polymerase, which will replicate the phage DNA. Now, does anyone see a problem here? Okay, the phage is trying to do two opposite things at once. It's trying to degrade DNA with the DNAs, and it's trying to polymerize DNA. How can that possibly work? Normally, it can't work, okay? You'll be making a new copy of your genome and it would just get cut up. So the way T4 phage gets around this is that it chemically modifies cytosine with hydroxymethylcytosine synthase, and DNA polymerase will incorporate hydroxymethylcytosine into the new copies of its DNA. However, the phage DNAs will not degrade DNA that contains hydroxymethylcytosine, so it will only degrade the host cell DNA. And then these nucleotides that are released will be used by the phage to make its own copies of phage DNA. So that's the way it gets around this problem. Now so once the phage DNA has been replicated, this is really the distinction between early genes and late genes in phage and in other DNA viruses. Early genes are expressed before the genome is replicated. Late genes are expressed afterwards. Now, late gene expression requires another early gene product, which is in Bacteriophage T4. It's not going to work, is it? I'm going to be doing this like forever. Okay, it's this protein GP55, which is a sigma protein. Everybody know what that is? Expression. You haven't done this in molecular biology? Okay. So, bacterial RNA polymerase is the core polymerase is a heterotetramer of these subunits, alpha 2, beta, beta prime. In order to be active, it's got to associate with a sigma subunit. The sigma subunit allows it to attach to a promoter region, and then this thing can carry on and transcribe. So different sigma subunits bind to different promoter sequences. So Bacteriophage T4 expresses GP55, and what you'll get is this polymerase. So these are all from the bacterium. So the bacterial RNA polymerase, this is from the bacteriophage, and it allows it to transcribe the late genes. So what do you have in the late genes? You have genes coding for the structural proteins of the phage, all the capsid proteins, and this allows the formation of virions, virus particles, and then finally the infected bacterium will be lysed, And that requires the expression, once again, of phage lysozyme and a whole-in protein. So the whole-in will form pores in the plasma membrane. The lysozyme goes out of these pores and will destroy the peptidoglycan. Okay, so that's how it works. Now, it's very important for phage T4 to express genes in the right order. So if you've got phage lysozyme being expressed right at the beginning here, the infected cell is going to explode before you've actually made any new infectious viruses. So it's this system to control early genes, replication, and then late genes, very important for the whole thing to work. And this is important here, okay? So this is what means that it's impossible for these late genes to be expressed until GP55 has already come up to a high level, okay? Because GP55 is necessary for the expression of all the late genes. Okay, I'm not going to ask this question because we have to move ahead with the timing, basically. Okay, so that's a lytic replication cycle. Bacteriophage T4 and all the myoviridae do this. Okay. Now, when people were looking at bacteriophage replication, 50, 60, 70 years ago now, it was found that some bacteriophage can infect host bacteria and not necessarily kill them. And these phage were called temperate phage. Okay, lytic, okay, you get infected, you die. Temperate phage, you get infected, you might not die. And bacteriophage lambda was one of these, well, I think probably the most studied temperate bacteriophage. Now, to visualize what's going on here, I'm going to show you some kind of video microscopy with a genetically modified bacteriophage lambda, which incorporates the gene coding for the green fluorescent protein in its genome. So the infected bacteria will be green, right? Okay, so that's what you're going to see. Okay, so you start off here, 2 bacteria have been infected. Oh, look, this one, dead already, lysed. This one, oh, it looks like it's quite happy. It's dividing, growing, just like all of its little bacterial friends here. Okay, so you have 2 very different fates of these 2 infected cells. This one over here got lysed. This one just carries on growing. So, okay, so here are the 2 originally infected cells. What is happening to this one? Oh, no, come back. Oh, I did the wrong thing. Oh, it's going through all the video again. Probably can't do anything until it decides to stop. Oops. That's kind of annoying, to be honest. Oh, yeah, it's come back now. Come back. Okay, forget it. Right, so. Okay, so what is happening to this one, right? So first, you've got your bacteriophage lambda, binds to the host cell, injects its DNA. First step will be to recircularize the genome. You'll have gene expression, so the infected cell becomes green. Then you have a lytic cycle. So DNA is going to replicate, get a lot of copies, produce a lot of bacteriophage, and then the whole thing will be lysed. So you have a lytic cycle like in bacteriophage T4. However, for this one, something different happens. Okay, so the first step is just the same. Phage DNA is injected into the host bacterium. It will recircularize. And then, instead of replicating, it's going to be integrated into the host cell chromosome. And this occurs by the action of the phage integrase, which recognizes a specific site in the bacterial host chromosome, the lambda attachment site. Now, once the temperate phage genome, or the lambda phage genome, has been integrated, Some phage genes can still be expressed, like the GFP here, but the genes involved in DNA replication, structural proteins, genes necessary to lyse the infected cell, their expression is repressed, shut off, okay? So what happens is, well, the bacterium just carries on growing, doesn't produce phage, and it doesn't get killed, okay? And the net effect is, in fact, every time the bacterium divides, it's going to replicate the phage genome along with its own genome. And so this is called lysogeny, okay? This is a lytic cycle. If my arrow is ever going to come back. Yeah, okay, this is a lytic cycle, and this is a lysogenic cycle. Why is it a cycle? Because this integrated form can be reversed. Under certain circumstances, the phage genome can be excised, and then it will restart a lytic cycle. So it's, lambda phage and other temperate phages are switching, okay? They have, some of the time, they're integrated copies in a bacterial genome. Some of the time, they are free, viral genomes in a phage particle. Okay, so general features of lysogeny. So in lambda, right? The decision between whether the phage is going to kill the host or integrate its genome occurs very early during replication. And there are two very early genes which are important, C2 and CRO. And depending on which one is dominant, then you get entry into the lytic cycle if you have too much CRO. Whereas if you have an excess of C2, then the virus will enter lysogeny, the phage will enter lysogeny. So the main thing is accumulation of C2. So what favors the accumulation of C2? Low temperature, low metabolic activity of the host cell, or if you have too many phages infecting the same bacterium at the same time. Each of them will start expressing C2, and there'll be enough to push the phage into lysogeny. And what happens afterwards is, so if you've got excess of C2, then the phage will start to express the integrase. So this initiates lysogeny, and then it will also express the C1 repressor. This is what is preventing the expression of the genes involved in the lytic cycle. So it maintains lysogeny as a stable state. Okay, so the integrated DNA is called a prophage, and the lytic cycle can be reactivated if the host cell is stressed. So this can be, heat shock, UV radiation, or some antibiotics can reactivate a phage lytic cycle. So yeah, so the thing, lysogenic phage equals a temperate phage. So these kind of conditions that favor lysogeny, what they have in common is that these conditions are not favorable for bacterial growth, okay? So if your bacteria are in the exponential phase of growth and you infect them with Bacteriophage lambda, there'll be a lytic cycle of replication because the conditions for bacterial growth are very good. So if the phage infects a bacterium and releases a whole bunch of other phage, there'll be plenty of bacteria around for these progeny phage to infect. So that makes sense. However, if you've got poor growth conditions for coli, for the host bacterium, then there's not much point in having a lytic replication cycle, because when you release all these progeny viruses, there won't be many bacteria around to infect. So it's a better strategy to just integrate into the genome, lie low for a while, and wait till the bacterium can start growing again, and then maybe reactivate a lytic cycle later. So that's why lysogeny exists, okay? Because it helps the phage employ two different strategies and adapt the replication strategy according to how the host cell population is doing. Okay, so just like everything in biology, it's complicated, but it's complicated for a reason. Okay. Right, so that's about all the things I wanted to say about, the bacteriophage replication. So the different steps in the lytic replication cycle in T4 and the lysogenic cycle in bacteriophage lambda. And then this last thing, why is it useful? Okay, why does this even exist, this complicated stuff? Okay, so last part here about bacteriophages is bacteriophage and human health. And there are really two parts to this. The first one is that prophage are part of the mobile genetic elements in bacteria that can carry virulence factors. Okay, so if you look at a lot of these pathogenic bacteria, some of their virulence factors are carried on integrated prophage. So, all of these exotoxins are carried on prophage, integrated into the bacterial genome. So E. coli, enterohemolytic E. coli, express Shiga toxins, which are in fact carried on a prophage genome. So these two prophages, SP5, SP15, which are here, code for these exotoxins, which makes this bacterium very, very dangerous. And this is also an interesting kind of like diagram, because you can see that, in this one genome, you've got a whole bunch of prophages there just hanging around coding for different stuff. Yeah.
So it means that without the action of the prophage, E. coli couldn't secrete this stuff.
Yeah, exactly.
So without the existence of the prophage, E. coli, that strain of E. coli will not be harmful for us, right?
It wouldn't be harmful, or it would have a different pathology. It wouldn't be so dangerous.
I mean, for this strain,
Yeah. So the virulence of this strain is directly related to the fact it's got these virulence genes on the prophage. So it's an important mechanism of exchange of virulence factors between bacteria. Okay, because the lysogeny is reversible. So, from time to time, these prophage can excise and be packaged into infectious phage particles and go and infect another bacterium. So this is one mechanism by which, virulence factors can be transferred from pathogens to just ordinary commensal bacteria. And this is what's called lysogenic conversion of a commensal bacterium to a pathogen, because temperate phage can transfer genes between different bacteria. And this is also possible between species, okay? So, E. coli can maybe pick up virulence genes from other gram-negative pathogens by prophage. So this experiment here, it showed transfer of genes between Listeria and Staphylococcus, two quite different gram-positive strains. So that's important. So that, it's kind of important for bacterial pathogens and virulence. So that's the bad part. And the good news is the use of bacteriophage as antibacterial agents. So kind of a bit of a kind of anecdote really, but it's something that still has potential. Okay, so Félix Derel, who's one of the co-discoverers of bacteriophage, was the first person to realize, these things, they kill bacteria. So maybe we could use them to kill bacterial pathogens and cure people. So that was his idea. And they actually did develop a lot of this stuff. And like in the 1920s, you could go into a pharmacy in France and buy some bacteriophage and like drink it. You could get this stuff. I mean,
so here we can just buy cocaine inside of
yeah, we're both at the same time, cocaine and bacteriophage. Okay, so this was kind of like a developing therapy until antibiotics were discovered. Okay, so now you have a small molecule that can work against all different types of bacteria. And so, that's it. Nobody's interested in phage therapy anymore after this. Well, until, of course, then we found that a lot more and more bacteria are becoming resistant. So this is the kind of emergence of multi-resistant bacteria has stimulated a kind of new interest in phage therapy. So there are some places in the world, yes?
Yes, I've got a question because I hear that apparently it?
Yes.
Because there is no resistance, but are we sure about it? For example, if we are giving phage or using phage or
Okay. So
in 50 years.
so the question is, how do we know there won't be resistance to phage? And probably we don't know, but one of the things about phage therapy is it's very specific. Okay, the phage that work, they will only work for a particular strain of bacterium, right? And often, well, what I've seen is that sometimes they use multiple phage, or several injected together in people. And whatever happens, you have to test the bacterium from a particular patient and find out the phage which are effective on that particular one. So it's a kind of very individualized therapy. It's never going to be like used as a mass therapy like antibiotics. So I think for that reason, the risk of emergence of resistance is lower because you'll just have less use basically. But biologically, sure, it can happen. Yes.
Is it possible that in the lysogenic cycle, some genes that we code for virulence factors that are inside of the bacterial genome could, by the transposon coming on the genome of the bacteriophage, and then maybe when it will be transferred, become harmful?
Yeah, that's also another point. So could some of these phage become temperate phage and exchange virulence factors? Only lytic phages can be used for this type of therapy. So that's a very important point. So you have to be sure that your phage is lytic and cannot become lysogenic. Okay, so getting back to what I was saying, so some countries or some places in the world carried on using phage therapy. So this is place in Georgia, that is like in the Caucasus, not the USA. So they've always used like bacteriophage ever since like the 1920s. So they have a massive collection of phage that they use for treating different stuff. And basically, on the outside, cutaneous lesions. So you've got like the infected tissue, the bacteria that you want to get rid of, they're, available. It's an open wound, this kind of stuff. And, there are some places in the States that use this. I was very surprised to find this. So there's this place in Texas. They also use bacteriophage to try and treat wounds on the outside of a person that are infected with bacteria and resist antibiotherapy. So there are a couple of places that do this stuff. Now, up until recently, I thought that this would never work for systemic infection. If you understand, it's on the skin, it's a wound. You can put the bacteriophage on there. You understand how it can work, right? But someone with a systemic infection, you think this is never going to work. But in fact, there have been some cases that have worked. So I'm just going to talk about a couple of case reports. So this one, 2016, 68-year-old man with a multi-resistant Acinetobacter baumannii infection. So this is gram-negative opportunistic pathogen. And this guy, he was almost dead, right? He was in a coma, no real improvement. And so I think it was his wife who got in touch with the scientists who are working on the research on this. And they managed to get together a kind of therapy with 9 lytic phages together with the antibiotics. And, they kept giving them, intravenously and intraperitoneally. And yeah, it really worked. So this guy survived. Kind of like miraculous survival story. So another one here, similar kind of thing, 2018. This man, 76 years old. He had a Pseudomonas aeruginosa, once again, gram-negative pathogen, in a kind of aorta graft, next to his heart. And they gave him 1 dose of this phage, OMK01, plus antibiotic, and it just eliminated the infection. Now, this one is really clever because it's got a very intelligent combination of the antibiotic and the phage, because the resistance mechanism of Pseudomonas aeruginosa against this antibiotic is to down-regulate the porin that allows it to get in. No, hang on, that's the wrong way round. No, upregulates the exporter. Okay, so he's got some kind of efflux pump that pumps out the antibiotic. So, and this efflux pump is actually the receptor for the phage. So if the bacterium is becoming resistant to the antibiotic, it's expressing more of the receptor for the phage. If it's becoming resistant to the phage by downregulating its receptor, it becomes more susceptible to the antibiotic. So this was really cool. They could this combination of phage and antibiotic. So, there are some of these, like this is early stage, right? One person, but the results are pretty positive and rather dramatic. So this is something that might be developed, getting more and more of these combinations, one particular phage with an antibiotic and try and trap the bacteria, okay? Okay, so kind of summary about this, characteristics of phage therapy. Okay, it's highly specific, all right? So they are narrow spectrum. You can't get one phage, it's going to get rid of all your Staphylococcus aureus. Every single person has got to get the bacterium, they've got to test which phage work against which strain. So it's quite labor intensive. The advantage here is, you won't like destroy your microbiome. Okay, the mode of action is interesting. So it's lytic replication. So that means that, as long as you've got the host bacteria there, the phage is going to amplify itself. So you might give one dose, but it can replicate and give yourself, more medicine locally. So that's kind of cool. There is no real cross resistance between antibiotic resistance and phage resistance. Okay, so lytic bacteriophage are effective against antibiotic resistant bacteria. One of the kind of downsides of this is that, if you've got a lot of gram-negative pathogens and you're going to go in and lyse them all, this was going to release endotoxin. So you might have a risk of a toxic shock with this therapy. Okay, and the particle, okay, so virion, it's a big, proteinaceous thing here. So it doesn't really have chemical toxicity, okay, so that's probably an advantage. But they are immunogenic. You could develop antibodies against them because they're proteins. And they've generally got a short half-life in blood. So you have to keep injecting them and people will develop antibodies and probably they won't work if you give multiple re-injections. So, it's not all fantastic, okay, but it might be useful in some situations. Right, so just to finish bacteriophage here, okay, don't forget we've got tons of phage in the biosphere. We've kind of important but mysterious roles in microbial ecology. Very important model system for molecular biology. Never forget CRISPR-Cas9. It's a bacterial defense mechanism against bacteriophage. And then, what has this got to do with human health? Lysogenic conversion of commensal bacteria into pathogens is by far the most important aspect here. Phage therapy, we're talking about, I don't know if it's even like 10 people per year in the whole world treated by this kind of stuff, okay? But it's very, very small numbers. Whereas this lysogenic conversion, this is going on all the time, in billions of people all the time. Okay, so that's it for this one. So I'm going to go on to, I can't see a thing on this screen because it's got too much sunlight. Okay, so we're going to move on to here, right? Yeah. So we're going to have a few kind of general considerations about human viral infections. And then what I'm going to do is I'm going to, oh, that's a bit weird. And I'm going to talk only about SARS coronavirus 2, because when I was getting into this, I thought, well, it's probably a good time to talk a little bit about the fundamental virology and then give an update about maybe the epidemic, treatments and vaccines. That's probably enough, right? I mean, if we could do other viruses, it's just going to be too much. Okay, so that's really weird because this blue stuff is generally red on my screen. It's very strange. So, okay, just general features of viral infections. So you can have acute and persistent viral infections. So most of the time when somebody says, oh, I was ill last week, I had a virus, you think about acute infections. And what happens is, you get infected here, the virus starts to replicate, then you have symptoms, and then you get better. Or you get infected, the virus starts to replicate, you get symptoms that get progressively worse, and you die. Okay? So a lot of acute viral infections are really quite harmless, adenovirus, rhinoviruses, common cold viruses. You can get infected several times a year, and like the infection fatality ratio is about 0 for these viruses. Everybody gets better, almost. Now, on the other kind of extreme, you have things like Ebola virus Zaire, which will give you an infection fatality ratio of about 60%. 6 out of 10 people that catch this will be killed. Rabies virus. If you don't get a very intensive medical treatment, you'll probably have like 100% death rate. If you get a good protocol of medical treatment, you have about 50-50 chance of surviving. And then in between, of course, you have all these other viruses, so influenza, seasonal and pandemic influenza. And this is SARS coronavirus 1, SARS coronavirus 2, with kind of fatality ratios that are not 0, but not really so extreme. So anything that is above about 0.1% is going to cause problems if it's a very transmissible virus. So those are acute infections. And as well as that, you have viruses that cause persistent infections. And persistent infections, there are really two types. So you have latent infections. So for latent infections, you have a primary infection here. The virus replicates, you get ill, and then you get better. But in fact, the virus is somewhere in your body in a latent state. Perhaps integrated into your genome, perhaps not, perhaps just as, yeah. Huh? Yeah, it's not a virus though. So sure, the malaria is like that, but that's because you have the hypnozoite in the liver, right? Which remains latent and then can be reactivated years and decades after the initial infection. So, herpes viruses are the champions of this kind of latent infection. Like if you have a zona, you have shingles when you're like 60, 70 years old. This is the varicella zoster virus that infected you when you were a kid, when you were 3 years old. It stayed in your body in latent state for decades and then can reactivate. Okay, then you have like chronic viral infections. So you get the primary infection, symptomatic, and then the symptoms kind of like decline, and then they can come back more or less, and then it kind of has a tendency to get worse and worse. But the difference between a chronic and a latent infection is in a chronic infection, you always have active virus replication all the time. So, so this is like the herpes viruses, the classical latent infections. Chronic infections, hepatitis B, hepatitis C. And things like HIV, they're kind of 50-50. If you don't have antiretroviral treatment, you have a chronic virus infection which will probably kill you if nothing happens. The reason HIV infection cannot be cured is because it's got the ability to integrate into the host cell genome and become latent. Okay, so that's kind of latent infection, or acute and persistent infection. So how do you get infected? Well, most viruses enter by the mucosa. So either, gastrointestinal, respiratory, or genital mucosa. And it's important to know this to kind of tailor the correct strategy for preventing transmission. So, we're never going to get anywhere to prevent COVID-19 by asking everyone to wear a condom when they have sex. That's not going to work, right? So you have to know the transmission mechanism in order to, use the right kind of preventive measures. Okay, the skin, pretty much a poor portal of entry for most viruses because your skin, this is made of like dead keratinocytes, right? There's no live cells here. So a virus has got to infect a living cell to be able to, start replicating. So the skin's a pretty good barrier against most viral infections. Okay, so there are some like papillomaviruses that give you, skin warts, and some pox viruses can infect just in a small like scratch on the skin. But most viruses that start their infectious cycle under the skin are injected by vectors, okay, by mosquitoes or by ticks that can bite you. And then they get access to living cells, subcutaneously. And of course, blood, some viruses can be transmitted by blood. So either by blood transfusion, intravenous drug use, or by organ transplant. Kind of weirder one, but it can happen as well. And the last one, which is very, very important for a lot of viruses, mother-to-child transmission. Either in the womb, in utero, this occurs for some viruses, or during childbirth, because you have blood from the mother, you have genital secretions from the mother that are in contact with the respiratory and the oral mucosa of the child that's being born, right, of the baby. So this can be a very important mechanism of transmission for viruses like HIV, hepatitis B, which can be at high concentrations in the blood, okay? Right, so that's, oh yeah, pathology, what else? Okay, so that's how the virus gets into the body. Once the virus has infected somebody, one of two things can happen. You can have a localized infection. So in that case, virus replication is limited to the initial site of infection. So you don't have viremia. Okay, so viremia in blood is what's that called? It's redundant. Okay, so viremia means virus in blood. So viremia in blood, it's rather poorly expressed here. So no viremia, and you have a short incubation time. So the incubation time is the period between you get infected and you have symptoms. Okay, so typically you're talking about, influenza virus or something like that. You get infected, you'll have symptoms within 2 or 3 days. Also true for things like, rotavirus, which causes, viral diarrhea. You get infected, you'll be very sick like 2 or 3 days afterwards. Okay, so local replication, no viremia, and short incubation period. That's local infection. Now, you can also have systemic infection. So this means that the target organ, that is the organ where you can detect symptoms, is distant from the initial site of infection. And the virus has got to get from the initial site of infection to the target organ through the blood. So you have viremia, and it takes longer for symptoms to develop. So it could be 1 to 3 weeks, or for some viruses, even like 1 month after infection, and then you develop symptoms a long time afterwards. So like typically, hepatitis A, okay, it's transmitted by mouth. The site of infection is in the intestine. It's hepatitis, the liver is the target organ. Measles, you might not think so, but, it's transmitted through the respiratory mucosa. You get a systemic infection, you get virus in the blood, and the symptoms are what? These big eruptions on the skin. And that will occur 2 weeks after the initial infection. The incubation phase is 2 weeks. HIV, genital mucosa is a site of infection. The target organ is the lymphoid tissue. Okay, so systemic infections. Okay, so now we're going to talk about, human coronaviruses. So we only got a half hour, so I'm going to probably go quite fast here, right? Okay, so as you all know, there are 4 strains, or up till last year, there were 4 strains of human coronaviruses, which caused seasonal infections. And they were grouped into 2 clades, the alpha and the beta coronaviruses. So, human coronavirus 229E, NL63, HKU, OC43. And then, ever since, well, for the last 20 years, there have been outbreaks of these new beta coronaviruses that have spilled over from an animal reservoir and started infecting people. And of course, the one that has really won the jackpot is the SARS-CoV-2. And so one thing you've got to be clear about here, the virus is SARS-CoV-2, so it's severe acute respiratory syndrome coronavirus 2, because genetically it's very close to the SARS coronavirus that was detected in 2002, 2003. So that's the virus. The disease is COVID-19. COVID-19 is not the virus. Okay, so a little bit about the fundamental virology of these viruses. Okay, so coronaviruses are enveloped RNA viruses with a positive strand genome. So the RNA codes directly for virus proteins. Got a helical nucleocapsid. And the important thing here is the spike protein. So these seem to form a kind of crown around the particle, and that's how the virus family got its name. And these spike glycoproteins are, for receptor binding and entry. And we'll talk about vaccines a little bit later on. So the whole idea is to generate antibodies against the spike protein, and these antibodies will be neutralizing and will prevent the virus from infecting you. Okay, so basically, it's a plus strand RNA virus. So what is happening is that the positive strand genome is going to get into the cytosol. Host cell ribosomes will translate some of the proteins here. And some of these proteins, or at least one of them, will be a virus RNA polymerase, which will allow the viral genome to get replicated. So it all happens in the cytosol. And then you'll have, release somehow. So what is happening? So for the attachment step, all right? So the spike glycoprotein is the attachment glycoprotein. The host cell receptors, well, they're different for the different coronaviruses. For SARS-CoV-2, it's the angiotensin converting enzyme 2, which is the same receptor as for SARS coronavirus. But it's also the same as for this, seasonal coronavirus. It's not just the receptor use that makes the virus so dangerous. Okay, the virus is bound. What happens next is it seems to be internalized by endocytosis, and then the spike glycoprotein has got to be partially degraded by proteases. And this will activate the fusion domain of the spike glycoprotein, which allows the genome to get into the cytosol. Okay, so once you've got the genome in the cytosol, what does the genome look like? Okay, so it's organized like this. You've got one large RNA molecule. And coronaviruses, they've got the biggest genome for any RNA virus. So they're kind of like champions for this. And if you look at it, like the 5' end has got two large open reading frames, and then the 3' end has got a whole bunch of different smaller open reading frames. And these ones here can be translated into two big polyproteins straight from the genomic RNA, whereas all the other 3' open reading frames have to be translated from shorter messenger RNAs. So how does this happen? Well, the first thing that happens is the genome gets into the cytosol, so the host cell ribosome is going to start translating it. And this will allow these two polyproteins, PP1A and PP1AB, to be produced. Polyprotein, what does that mean? That means you've got all these different proteins coded on the same mRNA. So when it comes off the ribosome, they're all joined together. And then they've got to be cut into the individual functional proteins. And these are the virus proteases that do that, the 3CL and what's the other one called? I can't remember. It's going to come up here. Oh yeah, the PL and the 3CL proteases. So the virus codes for its own protease, which chops this polyprotein up into the individual functional proteins. So 3CL is the major one. Then, okay, what do you have here? This is the RNA-dependent RNA polymerase. So this is the virus polymerase, which can synthesize a negative strand, okay? And then this negative strand is used as the template to transcribe these messenger RNAs. And then these messenger RNAs here, they encode for the structural proteins of the virus particle. You've got, you know, S, what is that down there? N, you've got the membrane protein as well, and they can package up the genomic copies and they're released from the cell. So 2 points here to remember. 2 enzymes necessary for virus replication, 3CL protease and the RNA-dependent RNA polymerase. Okay, how about pathology, right? So, you know, seasonal human coronaviruses, we probably had them loads of times in our lifetime. And generally, they cause seasonal respiratory infections, sometimes can cause pneumonia, can cause gastroenteritis. And these seasonal coronavirus, human coronaviruses, they're responsible for about 10% of the cases of children hospitalized for respiratory infections in France. So they're not entirely harmless already, these guys, okay? Now, it's not so easy to find studies of the general pathogenicity of these viruses because there haven't been so many community studies carried out, but I found one of them. So this was a study in New York in 2018, and they studied, children who had like respiratory symptoms in the community and in the hospital that was nearby. So if you're like you're a kid and you're catching one of these seasonal coronaviruses, most of the time, you get your nose is blocked up, you have a cough, fever, less frequent, okay, wheezing, a sifflement au poumon, almost nothing. So it's not infecting the lungs most of the time. And you'll be okay after 2 weeks, okay, for kids catching a seasonal coronavirus. The main thing is blocked nose and a cough. The more serious cases, you have fever, you have this wheezing, so there's some infection in the lungs. Okay, so compare that to the new beta coronaviruses, and the one that was discovered first was the SARS coronavirus in an outbreak in Hong Kong and the south of China in 2002-2003. Now, I say China and Hong Kong, there were limited outbreaks in other countries where people got on the plane and they went traveling. So this is data here from an outbreak, I think, in Canada. So SARS coronavirus, the incubation phase was 3 to 10 days with a median of 6 days. Almost everybody who got infected had a fever, dry cough, muscle ache, lung lesions, so you've got the virus infecting the lower respiratory tract, and 6.5% death rate. So this is very, very serious condition here. Yeah, so that was in Toronto. Now, when they broke down the death rate by age, for like all the cases that were studied in the whole of the outbreak, you got something similar to Covid-19. So among people less than 30, there's almost no risk of fatality, and the death rate got higher the older you were. So COVID-19 is something a little bit like this, okay? But SARS coronavirus, it looks like it's maybe about, 10 times more pathogenic than SARS-CoV-2. Okay, so SARS-CoV-2, what happens? Okay, so day 0, you get infected via the upper respiratory tract. Once again, the incubation phase apparently is 5 to 6 days on average. Minimum is maybe like 2 or 3, maximum 14. So that's why if you're a contact case, you have to wait 14 days. C'est cette histoire de la quatorzaine. And then what are your initial symptoms going to be? Fever, cough, fatigue, muscle ache. All of this stuff is quite non-specific. Could be a seasonal virus, could be influenza. And the only real specific sign is this loss of your sense of smell or taste. So this is really kind of like pathognomic, I think is what the clinicians say. Now, once you've started symptoms, then, okay, so this is a diagram from a paper earlier this year. So there's your incubation period, then you get these symptoms, and you've got another kind of period of 1 week. This is the second phase. You've got these symptoms, either they'll get better or they'll get worse, okay? So if they get worse, then you get shortage of breath and dyspnea. So you'll have trouble breathing. So of those people, some of them will need oxygen and some of them will need to be hospitalized and some will need invasive ventilation. And these percentages here, they really come from data from China that were published in March, April, okay? So what's it here? Yeah, Critical illness, 5% of the symptomatic patients. Severe illness, this is someone in hospital, 14% of symptomatic patients. That's from China. We're talking mostly about adults here. Now, how dangerous is this virus? That's not actually so easy, because certainly in the first phase of the outbreak, January, February, March, April, the people that the hospitals and doctors could see were the symptomatic people, right? And you don't really know how many total infections there were. So this is a paper that was published in July from a team of epidemiologists in the Pasteur Institute, and they tried to analyze all the data from France up till about May to try and figure out, how many people really were infected and what was the real rate of hospitalization and death amongst the population depending on the age range, okay? So these are the numbers they got. So the probability of being hospitalized if you're infected. Now for you young guys here, right, so it's like less than 1% chance of you being taken into hospital. Then as you get older, it becomes progressively worse. So if you're infected and you're like 80 years old or something, you're like 25% of chance of needing to be taken care of in hospital. And then this is the probability of death if you are hospitalized. Also, it's worse the older you are, right? So if you're taken into hospital and you're like 75 years old, probably had a 40% chance of dying. Now, one of the things they did with this, which I think was very good, was that they were able to calculate, or at least estimate, okay, what's called the infection fatality ratio. So that is, if you've got 100 people who get infected, how many people will die? And that includes the asymptomatic infections. So this is the number. So this is the number per age range. Okay. So basically, everybody younger than 30, the infection fatality ratio in France calculated to be like less than 0.01%. So if like the whole of the University of Nantes students got infected, we might have like 3 people die of this, okay? So it's like not very much really. Then, just like we saw for the SARS coronavirus 1, you have a higher risk with age. And then finally, this is the overall infection fatality ratio for the whole population of France. Now, this is going to be very dependent on the demographic structure of the country. We've got a lot of older people, retired people here, so we're going to have a larger number of serious cases. Some countries in Africa, a lot of the population is younger, so they're not going to have a lot of serious cases. So anyway, for here in France, they estimate is that we've got like a 0.5% infection fatality ratio. And that is a really important number because it determines what the worst case scenario is. You could imagine if 50 million people get infected in the country, that would be 250,000 deaths here. That's a significant number of people to be dying in 1 year, right? So this is that's the kind of like the size of the problem. Okay, so where are we in the epidemic? What's the current situation? So these are the numbers I got from our worldometers, which I like very much. It's a nice site. So here's the number of cases per day. That's like earlier this year, January, February, March, and then May here. And this is where we are now, okay, the number of cases per day. And this is the number of deaths per day. And one thing that's kind of really interesting about this is like earlier on in the year, we had loads more deaths. Not really so many cases. Now we've got a ton of cases and not so many deaths. So why is that? Well, right now we've got more testing capacity and we're picking up a lot more asymptomatic cases and this kind of stuff. So this number is a more accurate reflection of the number of real infections. This is an underestimate. The other thing is, maybe, the clinicians in the hospitals know how to take care of people a bit better. So the overall death rate is going to be a bit lower. And I think both of these things are true and both contribute to the kind of reduction in the death rate in relationship to the number of cases that we've seen. Wow, we've got 5 minutes. This is never going to work. Okay, so let's try and go through this quickly. Okay, so yeah, I'm going to talk about potential therapies here. So you have these different steps in the virus replication cycle that can be blocked by different treatments. So I'm just going to talk about some of these and go very quickly through some clinical data. Okay, so what definitely doesn't work, lopinavir, ritonavir, this is a protease inhibitor that works against HIV. And it also works in cell culture against the 3CL protease of the beta coronaviruses. So we thought, yeah, that's great. We could just use this. It will work. Well, in fact, it doesn't work. Okay, so first study in China, 100 patients, randomized in each group, no benefit. The UK consortium, once again, 3,400 patients with standard care, randomized against 1,600 for lopinavir or tenofovir, no benefit. Didn't work. And you get the same kind of thing for hydroxychloroquine. I know that Didier Alter says it's going to work, but in fact, here's a study from Brazil. They randomized patients, one third, didn't find any benefit. And this is once again the Recovery Consortium in the UK. 3,000 control patients in the control group, 1,500 in the hydroxychloroquine group, no benefit. So when people have tested these molecules rigorously, it doesn't really work. So remdesivir, okay, so this inhibits the virus RNA polymerase. Trump got this. Everybody in America is going to want this right now, otherwise they'll sue their doctor. So it's got very positive results in one trial here. Okay, so this was published September, maybe beginning of October. 500 versus 500. And in fact, you get people on the remdesivir arm got better a bit quicker. But there was no significant difference in the death rate, which was a little bit worrying. So just last week on Friday, there was a preprint of this paper from a big international consortium. So here we've got 5 times as many patients involved in the study. And here's the death rate here, okay? So this is the percentage mortality. Absolutely no difference between remdesivir and standard care. So this is telling us that there's no benefit of this therapy. So, not so great. Okay, monoclonal antibodies. I think I'm going to skip this because actually there's no data on patients with severe infection. So we don't really know if they're going to work. No trials done. Okay, so the other strategy to try and use is, all of this so far has been antivirals. Try to block the virus. But maybe to prevent people from dying, what has to be done is to look more about the pathological mechanisms. Why do people get killed? How does a virus kill you? Well, part of it is you have a kind of overactive immune response, which is going to give you a kind of shock syndrome. There seems to be excessive blood coagulation in the lungs, and this is called, kind of like a microthromby, and this is preventing blood circulation in the lungs. It stops the lungs from absorbing oxygen. And another thing is that you seem to have this cardiovascular thing where you have vascular leakage, pulmonary edema, so you have liquid getting out into the alveoli, and this is why you can't, ventilation doesn't work very well either, because people's lungs just can't absorb the oxygen. So the idea is perhaps you could use these therapies to try and improve the outcome here. So really the one that has been tested rigorously is dexamethasone here. So it's an immunosuppressor, okay? It's a steroid. So we got people randomized 4,000 versus 2,000. And in fact, was a significantly reduced death rate in the people the patients who got dexamethasone. Overall, not very big difference, but in fact, in the patients who were sickest, who were in the worst physical state, they had to have intubation and mechanical ventilation. Okay, so here, this is the patients who got the biggest benefit. So this I think really works, and everybody agrees about that. Okay. So it's not an antiviral; it's preventing the pathology. Okay, have we got time for vaccines in 3 minutes? Yes. Okay. Actually, no, we got 15 minutes, right? Okay, easy. Okay. You should have told me. I thought I was running. I thought I only had 5 minutes to do this whole stuff. I would have slowed down. Okay. Right. Yeah, take a big breath and slow down. Do you want me to go back and explain how some of this stuff is supposed to work or not? I got one for yes. Most people say no, right? Okay. Let's say no. All right. I'll just no. Yeah. And I think it's worth thinking how these things are going to work. Okay. So I can do the. Okay, remdesivir. I'll talk a little bit about this molecule. So basically, I didn't put the molecular structure on here. So it was developed as a therapeutic against Ebola virus. Okay, so Ebola virus is another RNA virus, highly pathogenic, and the molecule itself, it looks a little bit like a ribonucleotide. So the idea is that it's going to bind to the active site of the RNA polymerase, and it will be a competitive inhibitor. Okay, so a lot of so this is a kind of big drug family. I guess a lot of antiviral drugs are a bit like this. So, once it works against one RNA virus, you can try it against all other RNA viruses, and it might work a little bit. And so that's what happened here. So it works in cell culture, but it doesn't seem to be very effective clinically. So the chloroquine story is kind of a little bit different. So what is happening here is, okay, so during the endocytosis of the virus, the endolysosome becomes acidified. This activates proteases, and the proteases cut the spike protein and activate the membrane fusion to allow the virus to get into the cytosol. So the hydroxychloroquine can reduce the acidification of the endolysosome and should reduce the entry of the nuclear capsid into the cytosol. Now, one of the things that is a little bit interesting about this is that I'm not sure that's actually correct for all cell types, because there does seem to be a direct entry pathway for SARS-CoV-2, because it binds to this co-receptor called TMPRSS2, which is also a kind of protease which seems to be able to activate the membrane fusion activity of the spike glycoprotein. So in some cell types which express TMPRSS2, then the infectious entry doesn't seem to require endocytosis. So that might be one reason why hydroxychloroquine doesn't work very well actually in patients. Okay, because maybe down in the lungs where you have the pathology, the virus is not entering with this entry pathway, but rather by this entry pathway. And hydroxychloroquine can only have an effect if the virus entry is dependent on endocytosis. So that might be a reason why it's not working very well. Okay, monoclonal antibodies, I can do them then, if that weird. Okay, so monoclonal antibodies, how are they supposed to work? So they bind to the virus spike protein, so they can prevent attachment to the host cell. And so there have been a lot of great papers over the summer about getting monoclonal antibodies from infected people and characterizing them. So a lot of good candidates for this. There's no published data yet, okay, but there is a press release from the company, right? So they randomized patients, they got standard care, high dose or low dose of the antibody. But these were not hospitalized people. They were people who had mild infection. So all they could say is whether the viral load went down quicker with the antibodies or not. And it did, okay? So the viral load went down more quickly in these patients who got the antibody than in patients who just got standard care. Greater effect in seronegative patients. So if you're generating your own antibodies, adding some extra antibodies is not going to help very much. Okay, that's the message here. And, this is the big thing. Nobody knows if it will actually work on people who are seriously ill. Okay, so vaccines, right? So, now we just have 8 minutes. So vaccines, so the objective here, we've got to try and stimulate immunity and to prevent people getting infected in the future. Now, I found this great web resource here, and it told me that there are 213 candidate vaccines, 9 different categories, and there are 36 in clinical trials. So this is, massive vaccine effort. So what are the different types of vaccine candidates? So inactivated virus. You grow up the virus in cell culture, you inactivate it chemically with beta-propionolactone. You purify that, and you put it in a vial and you inject it into people with aluminium hydroxide as an adjuvant. So there are Chinese companies that are doing this, and also Valneva in Nantes. Their candidate vaccine is an inactivated whole virus. So what else have you got? You've got the recombinant protein. The whole point of these vaccines is to generate antibodies against the spike protein. So the recombinant protein idea is, okay, let's forget the virus. Let's just produce the spike protein, and you inject that. Okay, yeah.
But is it true that the antibodies of corona have a very short life?
Okay, the question is it true that coronavirus immunity is not long lasting?
Yeah.
Yes, that's true. So for seasonal coronavirus immunity, you don't have a long-lived immunity.
So does it mean, for example, for this vaccine, people should have to vaccinate themselves by
Okay,
2 months?
can I come back to this at the end? Because this is one of the big questions. Nobody knows the answer to it. All right, so right now, let's think, okay, imagine we want antibodies, okay? Think about whether they work later on. Okay, so the recombinant protein, just make the spike protein, you give that as the antigen. And then these other ones, they're kind of even more freaky than that. Say, okay, let's not bother to make the protein. Let's put the gene coding for the spike protein in another virus and inject that. So this is the recombinant adenovirus type candidate. So this is Oxford GlaxoSmithKline's candidate, and also, you know, the Russian one that Vladimir Putin got. That's a recombinant mix of 2 recombinant adenoviruses. And then there's, you know, even a more like space age sort of thing. Like, yeah, making a virus is too complicated. Let's just get the RNA, right? Chemically modify it to make it more stable, put it with a kind of protein that will package it, or a chemical that will package it up into a nanoparticle, and we'll inject that. So this is a company called Moderna. So real virus, protein, recombinant virus, RNA. So how do they work? Okay, so basically everyone's trying to get neutralizing antibodies. So just show a couple of results. So this is Novavax. So this is the recombinant protein produced in Bacteriophage, actually. So here's the results from this paper. So they've got one group of patients, or volunteers rather, they're getting placebo, they get nothing. And then they have different dosage schedules. And what they're measuring here is the neutralizing titer of antibody. So with, you know, protein on its own, high dose, you get some kind of like a response, pretty weak. But if you've got the protein plus their like special adjuvant ingredient, that really works. So that's after one injection, you get a reasonable response, and the booster injection gives you a really high neutralizing antibody titer. So how do I know it's really high? Well, this is what the neutralizing antibody titer you get in the serum of patients who have actually had the disease and recovered. Okay, so the amount of antibodies you're getting from the vaccine here is as much or even more than you get after getting really infected. Okay, so the vaccine can induce an immune response that is as strong as if you got infected. So that's good. Now the other question is, okay, the people who really need the vaccine are people who are over 65, who are over 70. And this is a population which generally is difficult to vaccinate. So responses against the influenza vaccine in older people are generally not so good. So this is the Moderna, the RNA vaccine. And they tried, you know, their candidate vaccine, low dose, high dose, with people who are, you know, more than 70 years old. And this is their neutralizing antibody titers. They're pretty good, okay? So once again, they've got these convalescent serum from patients who've actually been infected, and it's about the same level here. So you've got a good immune response in the high-risk group. So here we go. This is my summary. Now I really do only have 3 minutes. All right, so right now, the way I see it is we're in the middle of a very serious second wave in the Northern Hemisphere. It's not just France, it's the UK, it's other countries in Europe, and, you know, the US is not doing really very well either. So we're in the middle of this really very serious second wave. You have to remember the infection fatality ratio. The estimate is about 0.5% for the French population. So the absolute worst case scenario we're talking about is, you know, could have about 250,000 deaths. And this will definitely be like more than 1 million in the States. You know, this is the potential. The virus has got the potential to kill that many people. Unfortunately, so far, for all the antiviral therapies, they haven't had a massive impact on mortality. So there's no miracle cure, okay? But, treating the pathology seems to be what works better to prevent these very severe cases from killing people. However, so looking a bit further in the future, there are many good vaccine candidates. But, the question is, will they really prevent severe infections? They can produce antibodies. We won't really know until much later whether they've prevented deaths. And, it's a question. Oh, yeah. And the other question is that, what you said, will it really last a long time? Because for seasonal coronavirus infections, people get reinfected year by year. Okay, so the best explanation I had from this was it was on a podcast called This Week in Virology. And they had a big coronavirus immunity expert on there. And what he said was that, with the seasonal coronavirus infections, it's restricted to the upper respiratory tract. And here, immune memory is not really long lasting. So you get reinfected, but you control the infection very quickly. And his hope and feeling was that, once you get the vaccination, you get IgG. IgG is in the blood, right? It's not out here in the nasal mucosa, the upper respiratory tract. So these vaccine candidates might not actually prevent the virus from circulating. I think that's not going to happen. This is going to be the 5th human coronavirus. It's here. It's going to be here forever now. So that's why I think the question is, will it prevent severe infections? So does the fact that you've got high antibody titer and a memory T cell response, is it going to prevent you from progressing from mild symptoms to severe symptoms? And I think it could work, but we don't know, all right? Nobody will know until next spring, I think. So it could work because your immune system, will have a bit of an advance on the virus. And the other big question, will they be available in time? I think that's the big question here, because right now we've got tons of infections. If the vaccine becomes available too late, it's not going to help very many people. We'll all have been infected and either survived or died. Okay, so I'll leave you with that and carry on being careful with your coronavirus stuff.